Inside every living cell lies DNA, the blueprint of life. DNA contains genes, the instructions for building and maintaining an organism. But how do cells know which genes to read and when? The answer lies in special DNA sequences called promoters and consensus sequences. Imagine a vast library filled with books or genes. Promoters act like librarians, guiding the cell's machinery to the right books or genes at the right time. Consensus sequences, on the other hand, are like the signs on the library shelves, providing specific instructions to the librarians. Promoters are specific regions of DNA located upstream of a gene. They act as landing sites for proteins called transcription factors, which help initiate the process of gene expression. Think of promoters as on switches for genes. When a transcription factor binds to a promoter, it signals the cell to start transcribing that particular gene. This process ultimately leads to the production of proteins, the workhorses of the cell. Without promoters, genes would remain silent and the cell wouldn't be able to function properly. Consensus sequences are short, recurring patterns of nucleotides, the building blocks of DNA found within promoters. These sequences act like a specific language that transcription factors recognize and bind to. One well-known consensus sequence is the TATA box, named after its sequence of nucleotides, TATA-A. When transcription factors encounter the TATA box within a promoter, they recognize it as a signal to initiate transcription. Different consensus sequences attract different transcription factors, allowing for fine-tuned control over gene expression. Understanding promoters and consensus sequences is crucial for unraveling the complexities of gene regulation. Mutations or alterations in these sequences can disrupt gene expression, potentially leading to diseases like cancer. By studying these regulatory elements, scientists can gain insights into how genes are controlled and develop new therapies for genetic disorders. Furthermore, manipulating promoters and consensus sequences has become an essential tool in biotechnology, allowing for the production of valuable proteins like insulin in bacteria.